Welcome. Today we will explore evolution from a modern genetic perspective. Let's start. Once upon a time, evolution was simply defined as the gradual change in living organisms. While this definition is not incorrect, it is far too vague to serve as a foundation for building mathematical models. What exactly is changing in organisms? Over what time scale? At what rate? And how can this change be measured? These crucial questions cannot be answered by the old oversimplified definition. Hence, modern evolutionary biology adopts a much more precise definition. That is, evolution is the significant change in the allele frequencies within a gene pool of a population from one generation to next. Again, evolution is the significant change in the allele frequencies within a gene pool of a population from one generation to the next. But what exactly do we mean by population, gene pool, and allele frequency? A population refers to a group of individuals from the same or closely related species capable of interbreeding and producing fertile offspring. Typically, these individuals inhabit the same geographical region. For instance, imagine a forest inhabited by a group of red fox, Vulpes vulpes. They all belong to the same species and can interbreed, exchanging genetic material through their offspring, since they live and interact within the same environment. This group of foxes constitute a population. Keep in mind that if a geographical barrier such as a mountain or a river separate this group from others, gene flow between population may be restricted. Over time, this can lead to a genetic divergence, say between mountain fox and plain fox. Gene flow or migration means the movement of individuals into or out of a population can certainly alter the gene pool and allele frequency. Thus, a population is not just a group of similar organisms but a dynamic genetic system interacting with the environment. Let's examine the term gene pool. It encompasses all alleles of all genes across every individual in a population. To truly grasp this concept, we must understand genes and allele. A gene is a functional segment of DNA composed of a specific sequence of nucleotide like ATGC. It is transcribed into RNA, which may be translated into protein such as hemoglobin gene, hemoglobin protein, or function as non-coding RNA like rRNA, tRNA, which plays role in protein synthesis and gene regulation. Genes are inherited from parents and often determine observable traits like called phenotypes such as eye color. On the other hand, an allele is an alternative version of gene that arises due to change in its DNA sequences. Alleles are located at the same locus on homologous chromosome. They arise from the mutation. Even a single base pair change can create a new allele. Diploid organisms carry 2 alleles per gene. But a population can have multiple alleles, such as ABO blood group. Now, next we explore the allele frequency. The allele frequency refers to the proportion of a specific allele relative to the allele of that gene in population. It is essential to distinguish between allele frequency, count of a specific gene version, like suppose R and capital R and small r, capital R equal to red, small r equal to white, where genotype frequency, count of a specific combination like capital R or small r. Change in allele frequency drive evolution, not change in individual trait alone. For example, if the R allele become more common, red flowers may become more prevalent in the population. There are 4 primary mechanisms of evolutionary change. One is mutation, that means random changes in DNA introduce new allele. Migration or gene flow, movement of individuals alter gene pools. A random genetic drift in a small population, chance events can shift allele frequency. And number 4 is natural selection. Advantageous allele become more common because they increase reproductive success. Each of these can cause allele frequency to shift from generation to generation if none of them occur. If the population reaches a theoretical equilibrium, the Hardy and Weinberg equilibrium assumes no new mutation occur, no individual migrate, no genetic drift happen. All individuals have equal mating chances. All alleles have equal survival and reproduction chances. Then allele frequency remain constant and no evolution occur. This state is called the Hardy-Weinberg equilibrium, named after British mathematician Godfrey Harold Hardy and German physician Wilhelm Weinberg. They formulated the mathematical principle underlying genetic stability in a population. If real world data violate this equilibrium, it indicates that evolution is occurring. The Hardy-Weinberg formula help us qualify evolutionary changes and identify which evolutionary forces may be acting on a population. By the way, let's come to know about the directional versus non-directional evolution. In case of directional evolution occur when allele frequency change in a predictable direction, often due to consistent environmental pressure or migration. On the other hand, non-directional evolution caused by random genetic drift happens when changes are unpredictable, especially in small population. The question is, why do allele frequency fluctuate at all? Why do allele frequency fluctuate at all? Darwin and Wallace first proposed that variation among individuals leads to differing survival advantages. Over time, advantageous traits and thus their corresponding allele become more frequent. But where do new allele come from in the first place? The answer is mutation, a raw material of evolution. While mutations are random, natural selection filter them based on survival and reproductive fitness. In short, mutation provide variation, selection refines it. This interplay drives the evolutionary journey.